Okay, so I'd like to welcome everyone to the first of our three-part webinar series in celebration of International One Health Day. So I'm Dr. Cassidy Rist, an assistant professor in the Department of Population Health Sciences here at the Virginia Maryland College of Veterinary Medicine. I'm the faculty advisor to the Student One Health Working Group, which is made up of about 12 students representing all of our college's degree programs, including veterinary medicine, public health, and biomedical and veterinary sciences. So our working group also has student representatives from Virginia Tech Carilion School of Medicine and the Edward Via College of Osteopathic Medicine. The purpose of the student working group is to host events and activities like this one that support communication, training, and learning across human and animal health professions. So in today's webinar, our panelists will be discussing the role of biomedical research in developing therapeutics for COVID-19. I'd like to go ahead and introduce our moderator for this session, Juselyn Tupik, who is a member of the One Health Working Group. Juselyn is also a second-year dual PhD DVM student in Dr. Irving Coy Allen's lab in the Biomedical and Veterinary Sciences program. She's studying NOD-like receptor immune system recognition of bacterial infectious diseases, including brucellosis and Lyme disease. She hopes to integrate her research and veterinary training to become a veterinary clinician scientist. Juselyn, take it away.
Thank you for that introduction, Dr. Rist. I will begin by introducing the panelists. So our first panelist is my advisor, Dr. Coy Allen. He is an associate professor in the Department of Biomedical Sciences and Pathobiology. His lab is focused on exploring the intersection between immune system and host pathogen interactions, specifically understanding the contribution of unique families of cell receptors that modulate inflammation. Of particular relevance to SARS-CoV-2, the Allen Lab research team is studying a receptor that they believe functions to attenuate the so-called Cytokine Storm and the multi-system inflammatory syndrome that occurs in severe COVID-19 patients. Thank you, Dr. Allen, for being here. Our next panelist is Dr. Nisha Dougal. She is also an assistant professor in the Department of Biomedical Sciences and Pathobiology. Her lab studies evolution, transmission, and pathogenesis of emerging viruses. Her lab's work on SARS-CoV-2 thus far has centered on developing animal models of infection, testing antiviral compounds in vivo, and testing disinfection processes in vitro. Thank you, Dr. Duggal, for being here. And our last panelist is Dr. James Weger-Lucarelli. He is an assistant professor in the Department of Biomedical Sciences and Pathobiology. His lab is pursuing several lines of SARS-CoV-2 research, including the impact of obesity on disease following SARS-CoV-2 infection, developing molecular tools to study SARS-CoV-2 genetic determinants of human infection and transmission, and developing novel therapeutic and antiviral strategies to prevent COVID-19 and SARS-CoV-2 transmission. So thank you to all of our panelists for being here. First, we're going to start with a brief introduction for all of the panelists' research on SARS-CoV-2, starting with Dr. Allen, followed by Dr. Duggal, and then Dr. Weger. So take it away, Dr. Allen.
I don't know where else to go. You introduced me so well. So welcome, everybody. Again, I'm Coy Allen. I'm an associate professor here at the vet school in the Department of Biomedical Sciences and Pathobiology. The work we've been doing with SARS-CoV-2, which Juselyn mentioned, we're studying families of pattern recognition receptors. These are the receptors on and within cells that recognize damaged pathogens and stress. And our lab has been studying how these receptors function in the recognition of SARS-CoV-2. And then following recognition, these receptors trigger cellular responses. And so we've been trying to understand these cellular responses that get activated and how they drive inflammatory processes, and ultimately how these receptors help defend the host against SARS-CoV-2. And in some cases, how these can modulate the inflammation that occurs following SARS-CoV-2 to help modulate some of the inflammatory diseases or some of the inflammatory presentations that we often see in COVID-19 patients.
Thanks, Dr. Allen, for that introduction. Dr. Duggal, you can give your overview.
Sure. So my lab traditionally studies mosquito-borne viruses, and we're primarily interested in evolution and transmission of those viruses. So we've adapted our sort of general principles of research and studying SARS-CoV-2 in similar ways. So we're interested in transmission, and typically to do that, we develop animal models. So we set up a mouse model of SARS-CoV-2 infection here using a transgenic mouse line, and we're just got some funding to set up a hamster model, which I'm super excited about, which is really nice because hamsters actually transmit between each other. So it's a really nice system for studying transmission. The mouse model, on the other hand, isn't so good for that. Anyway, so we're studying transmission. We've also done some work with some local companies looking at their equipment that they're developing for disinfection of sort of large surfaces. And so we've had some work on that, looking at UVC and activation of SARS-CoV-2.
Great, thank you. Finally, Dr. Weger.
Yeah, thank you. So yeah, like Dr. Dougal, my lab normally works with mosquito-borne viruses, but after the pandemic hit, we started adapting some of our previous expertise towards trying to tackle this important problem. So the first thing is trying to test novel antivirals against SARS-CoV-2. So we collaborated with several groups here at Virginia Tech and some external groups as well, trying to see if we could develop some molecules that would be effective either before infection or during infection to prevent severe disease. And then another thing that we're interested in is how obesity or other metabolic conditions affect disease outcomes. So I think you guys probably all know that obese and diabetic people have worse disease outcomes than normal weight people. And so we're trying to figure out why that might be, and also how obesity or diabetes might influence transmission of the virus. And then finally, it's kind of a wide range of interests. We are interested in how you manipulate these viruses genetically in order to determine how they're changing over time. So identifying mutations that have arisen in the virus that might have conferred increased human-to-human transmission, and then trying to determine what those mutations are and what they might be doing to allow more efficient human-to-human transmission. So.
Great, it sounds like we have a lot of diverse research happening here at the College of Veterinary Medicine. So for the next part of this webinar we have individual questions for each panelist, and then we will have some general questions that any of the panelists can address. First, starting with the individual questions for Dr. Allen. What do you believe are the most concerning factors of SARS-CoV-2 virulence that we have yet to elucidate the mechanisms for? And what role do you believe biomedical research plays in uncovering these mechanisms?
Yeah, so that's a great question, Juselyn. So I'm an immunologist. My background is actually in respiratory viruses and the immunological impacts that they have. And so with this, I really have two concerns. I don't necessarily know if they're truly virulence concerns, but in thinking about the viruses, I really am concerned, at least with SARS-CoV-2, about the cytokine storm, which we've mentioned a few times. This has been reported in several COVID-19 patients. Normally, inflammation is a good thing. It's part of our natural response to damage, pathogens, and cellular stress. But following SARS-CoV-2, the inflammation in some of these patients really can't shut down. And this can cause a hyperinflammatory response. And ultimately, this inability to control overzealous inflammation causes a lot of collateral damage in the lungs. And we see this in a lot of SARS-CoV-2 patients, a lot of COVID-19 patients. And really understanding how and why this occurs in some patients and not in others is really important. And this also goes along with why some patients are display or are more, they have more pronounced symptoms than others. So some patients may be asymptomatic, others can be deathly ill. And so we think that all these factors seem to be correlated or at least related to each other. And we don't understand really why or how that is. And so that's one of my biggest concerns with this virus, that really we don't understand this quite yet. And then also, again, as an immunologist, really the strength or the ability of us to generate a potent immune response following SARS exposure or following a vaccine is really concerning to me. It's not really clear yet how strong of an immune response these vaccines can generate, or how long lived these exposure to viruses can at least the immune response to these viruses will last. And so understanding the ability of us to vaccinate and generate a really strong immune response to vaccination and previous virus exposure, I think is also really concerning. There's some data suggesting as little as 3 months of protection. And so it really is important for us to understand why and how to best vaccinate and what to target in terms of the virus and in terms of vaccine development. And so what role will biomedical research play in all this? I mean, I think the world is learning that we as biomedical researchers are really important. I think almost all of the discoveries that we're talking about, all of the progress that's been made, it's been an amazing amount of progress in a really short amount of time, has really been driven by our biomedical infrastructure, by our biomedical researchers at both academic, government, and industry levels. And I think this has really driven the innovation that we're seeing. It's really driven the progress that we've made. This has really been, it's a historical time, and we as biomedical researchers are certainly making some amazing progress. And so it'll be interesting to see over the next year or so, the vaccines that come out, the treatments that are developed, and literally, I believe the other panelists and I will agree that new research is coming out multiple times a day, all from harnessing our biomedical infrastructure here in the US and globally.
Yeah, thank you, Dr. Allen. That's actually a really great segue into our next question for Dr. Weger about vaccine development. So our question is, when developing a vaccine for SARS-CoV-2, what types of vaccines do you believe are the most promising candidates? And how should government regulation of vaccines be enforced during a pandemic, just with the expedited timeline through moving through phases of clinical trials?
Yeah, it's a really good question. And I mean, like Dr. Allen said, I think the concerning thing is that people are not really developing a great immune response even when infected with the actual virus. So how do you develop an effective immune response from a vaccine? And so, and also like Dr. Allen said, it's hard to keep up with the literature of all the vaccines that are coming out. I just looked, there's at least 193 candidates. And because of the sped up timeline, or maybe just a change in the way vaccines are being developed, most of them are either, so I don't know how much the attendees know about vaccines, but most of them are either a DNA vaccine, so that would be a plasmid where it expresses some SARS-CoV-2 protein inside the body as an immunogen. And then you also have viral vector vaccines, so using an attenuated virus to express some SARS-CoV-2 protein, so kind of a similar concept. And then the other one that's being commonly used is an mRNA vaccine, which is basically the same concept, is some way to express an immunogenic portion of the virus. But what you're missing in all those approaches is the rest of the virus. This virus is very large, as a 30 kb genome, and you're really only giving sometimes maybe 10% or less of the genome. So you may develop a strong antibody response with these vaccines, but you might not develop a very effective T cell response, or it might be very short-lived. So to answer the question more directly, I think that a live attenuated vaccine, so that's a weakened version of the virus, would be the most effective. But that takes a long time to develop. There are greater safety concerns. And so with this Operation Warp Speed, these other vaccine modalities are probably the most likely to succeed in a short period of time. But there is a concern as of how immunogenic they are. And with SARS-CoV-1 and MERS, other coronaviruses, it's known that if there is a poor immune response developed to vaccination, it can actually enhance disease. So there is some concern that on a greater level, a subset of people that are vaccinated will actually get worse disease. And so with these things going through so quickly, I guess this goes into the second question of how government should regulate that, which is a question that's way above my pay grade. But I feel like they should be very cautious. I know we all want to get back to eating at restaurants and whatnot, but not at the expense of vaccine adverse events. And we all know that vaccines are already struggling to maintain a good reputation because of all the misinformation. And so if there was to be some large adverse events that occurred from a vaccination, I think that could really hurt people getting other vaccines like the measles and other vaccines that people really need to be getting. So I think that the government should proceed cautiously and only approve a vaccine that's actually shown to be effective and to not have too many adverse events.
Yeah, I think you raised some really great points to consider. I know that's something that we're definitely all worried about, is getting a vaccine that's safe for everyone out there. Thank you. So kind of moving aside from vaccines, a question for Dr. Dougal. What are other therapeutics that are important for controlling COVID-19? And I know that you mentioned in your research that you were looking at some therapeutics, and what are therapeutics that you know for research in general that are being developed right now for COVID-19 besides vaccines?
Sure. So vaccines are excellent for preventing infection is the goal, but for individuals who are either can't get the vaccine or have gotten the virus and are ill, there are therapeutics that can be used to either prevent infection or to slow down disease progression. So there can be some therapeutics that are used prophylactically for individuals who can't be vaccinated in time. And that can be used in some situations where there's a known exposure. That would be sort of a limited use, I would say, of a therapeutic that would be developed. More likely, a therapeutic would be developed to treat disease symptoms after someone has gotten sick. And there can be a couple of different kinds. There can be ones that directly target the virus and prevent viral replication. So antiviral drugs. And there can also be drugs that treat the sort of downstream effects of the virus replication. So preventing the cytokine storm, for example. And so there are a lot of drugs that are known to blunt immune responses in relation to other diseases, autoimmune diseases or other infectious diseases that are trying to be repurposed for COVID, and those are getting fast-tracked if they've already gone through some sort of safety data processes. For other diseases, they can be tested more quickly against in COVID patients. So that's probably the fastest way that we're going to get a therapeutic, is something that's designed to prevent inflammation for some other purpose that can be used during COVID. Ideally, though, the best, in my mind, therapeutic would be something that would be antiviral. The reason is that not only would it prevent disease in an individual, but it could prevent transmission as well. So if you're actually reducing the amount of virus, then you can also prevent transmission. So that would be a nice added benefit of an antiviral. But we know that SARS-CoV-2 is an RNA virus, and so there's going to be a concern about drug resistance. So influenza, for example, I think I just checked and there are now 4 licensed antiviral drugs for influenza. And there is some amount of drug resistance in different areas that is slowly accumulating. And so that means that if you go with the antiviral approach, that there has to be more than one, potentially used in combination. So it does get complicated, but I think there's definitely, seems like there's different approaches for developing therapeutics.
Great, thank you. So now we have some questions to address for all of the panelists that I'm sure a lot of the public is really interested to have some answers to. Our first question is, why is COVID-19 causing such different clinical presentations in people even within the same family? I know that I've heard stories myself where one family member will be almost hospitalized level of sick and other family members will be asymptomatic. And I think a lot of people are very confused about why, like within families who are very genetically related, why we're seeing different clinical presentations.
I mean, I'm happy to start out. I mean, you mentioned genetics. Genetics plays a big role. And also there's a lot of heterogeneity, or there's a lot of differences in how the immune system in each individual recognizes different aspects of any pathogen. And so how subtle differences in each individual's immune system recognizes and functions can drive very significant differences in how those diseases present. And so certainly, and I'll let James and Nisha discuss also their perspectives here, but I'm sure James will mention something about obesity and lifestyle, things like that. It can all have a significant factor.
Yeah, for sure. I mean, there can be a lot of diversity within families in terms of metabolic status. Obviously, age is a big one. So, the older you are, the more likely you are to have more severe disease. I'm sure Nisha will mention some other risk factors as well, but one for me is the big one is obesity or just metabolic status. It could be diabetes or pre-diabetes, and it's not really clear why these conditions result in more severe disease, other than maybe this, like Dr. Allen mentioned, the overzealous immune response being even more overzealous in these people and therefore causing more severe disease. So.
There's also thought to be sex-specific differences in disease as well. So men, of COVID patients who are hospitalized, there's a greater proportion who are men. And it's for SARS-CoV-1, it's been shown that estrogens can play a big role in disease outcomes. So I believe that there are clinical trials right now looking at estrogen therapies in individuals for affecting disease outcomes. So I think there's, it seems like there's a lot of genetic, biological, and also lifestyle differences that can impact disease.
I've also recently seen some work where the actual dose, the initial inoculum, the initial dose that you receive actually can play a role in the severity of your disease outcome and course. And that kind of makes sense. If you just get exposed to a little bit, you don't get all that sick. But if you get exposed to a lot of virus all at once, then you tend to have a more aggressive disease course. So that's a good plug for wearing your mask.
Certainly, and I'm kind of curious whether there's differences in disease outcomes based on the mode of transmission. I don't actually have any evidence of this currently right now, but so the virus can be transmitted either by contact or non-contact mediated transmission, and that it could be related to dose. It could be that there's different dosages during different transmission routes or something like that that could impact disease severity, but I'm not sure I've seen any good evidence on that yet.
Yeah, and kind of continuing with the dosage question, what do you think is the most effective way of transmission? Like, are we talking about contact on surfaces or airborne as if you're talking to another person without masks? I think I've had a lot of friends ask me questions about things like that, so it'd be good to have some answers.
Yes, to all of it. It's fomites are traditionally a great way to pass viruses around. My research and my area of expertise is really in the respiratory route, and so I tend to think that's the more predominant way. But there certainly is strong evidence suggesting, and Nisha and James may know this, but I'm not sure how long the virus actually lasts on surfaces. Nisha, I think you're doing some work on that. But you can absolutely pick it up off of surfaces as well, and that's a significant driver of this. So yeah,
for sure, it does last on surfaces and in the air for quite some time. It's thought to be a little bit more stable than SARS-CoV-1 as far as sticking around, and so I think all of these things are potential mechanisms of transmission. And I think one thing to consider too is that transmission is very efficient. It appears from asymptomatic individuals. So typically we think of respiratory droplets from coughing and sneezing, but for asymptomatic individuals, those aren't necessarily happening. And so one possibility that my work is hopefully going to be involved in is looking at the transmission of the virus just through exhaled breath in the absence of, sort of the large droplets that come from coughing and sneezing. So I think there's possibilities of all of those happening.
Great, thank you for your responses on this. Moving on to a different sort of question, one that's definitely relevant to all the panelists here, is to what extent has your research benefited from interdisciplinary and outside collaborations? I know that I personally worked on projects with all three of you, and I'd love to hear your opinion on how collaborations are working on this. Whoops, wait.
Yeah, so yeah, like you mentioned, Nisha, James, and I all have collaborative projects both amongst ourselves and with other folks all over the campus at Virginia Tech. One of the reasons why I came to Virginia Tech was the collaborative nature of the university itself, and that's been a big help in my research. For us personally, in terms of SARS, we have several folks from outside disciplines coming to talk to us. For example, we have individuals from engineering coming to talk to us. We have folks from chemical engineering. James and I know we're both working on a project looking at different coatings for coating surfaces that have shown some promise as anti-SARS materials. We are also working, my lab itself is also working with a group in engineering to make a COVID-19 on a chip model, which is basically growing lung tissue in sections on different chips to allow us to rapidly screen drugs. And so having some of these really neat transdisciplinary collaborations is really, it's gonna really benefit, I think, the science and move the field forward. And yeah, I'm sure Nisha and James both have some similar collaborations as well.
Certainly, I think Virginia Tech has done a good job of promoting interdisciplinary work, specifically with SARS-CoV-2. There's been a couple of internal calls for projects related to this research, I think, and they've really done a good job of selecting people from all different fields so that there's lots of different ways of addressing the questions. And so I think this has certainly given me the chance to work with people that I would never have gotten to work with before. So, which is really exciting because we're all new. The great thing about it is that we're all new to SARS-CoV-2, so we're all learning together. So I think, yeah, it's been a good chance for me to work with lots of folks who I haven't worked with before.
Yeah, I feel the same way. I was approached by several people from different colleges, from, like Coy said, from engineering, and people with different compounds that they had previously tested for other conditions, be it cancer or antibacterial compounds and, surface coatings and things like this. And not everything worked out, of course, but that's science. Some of the things did work out. And just creating relationships with people that probably I would not have been connected with has been really nice. So it's a really nice collaboration. We're all working hard to generate some data that will produce hopefully some significant impacts on the society at some point. So it's been exciting.
Continuing with that question, you guys mentioned collaborating with people even in different fields outside of biomedical medicine. So what do you think is the ideal dream team as far as getting researchers together besides just people looking at biomedical research?
At least for me personally, it's less about the disciplines and more about the team itself. So it's certainly, I mean, disciplines certainly matter, but it's really about blending ideas. So diversity really matters when we talk about, and teams coming together to work together that are very diverse in their ideas and really in their way of thinking. That's one of I think when I'm forming a dream team, as you said, I really look for people who have some really cool ideas and can think about problems in a different way than I think about problems. By having several people thinking about the same problem, but coming at it from different directions, I think it can really drive solutions. And that's really, I think it's less about the different disciplines that are coming together and more about the different ideas. At least for me, it's more about the different ideas and the more about thinking problems through more logically and from different directions. There's so many people out there that are way smarter than I am, and I like to have them on my team. They make me look better. So. Mm-hmm.
That's great. Yeah, I know personally that I've benefited from collaborations within other labs to do my own research. So I'm glad that we're seeing that through COVID-19 research. Our next question is addressing basically what we want the general public to take away from COVID-19 research. I know that a lot of research kind of gets lost with the medical jargon. Like I know that Cytokine Storm has come up. A lot of my family members, they have no idea what that means. And I didn't know what that meant a year ago when I first started this program. So what do you believe the scientific community can do to better communicate research findings in layman's terms to the general public?
As far as the first question, what can the general public what can they take away from the ongoing research? I think the big thing is how impressive it's been that people from very diverse disciplines have been able to shift their research entirely towards one topic of great medical interest right now. And I was actually thinking about this the other day, that if, in a year when this is all over, hopefully, knock on wood, if we could do this like towards malaria or dengue or some other infectious disease that has a great significant medical interest, or that we could make a lot of progress very quickly. So I think that that's been one thing that the scientific community is able to adapt and produce a lot of really good science in a very short amount of time that can have a good public health impact.
I also think hopefully the public will have a better sense of what it takes to do this kind of research and why it's good to do some of this stuff for viruses that haven't caused pandemics yet. That's what I'm hoping also will come from this is, the non-scientists are learning a lot about virology and immunology and vaccines and all kinds of things that maybe they didn't know about before, and sort of understanding how we're working on these questions all the time. And I hope maybe there will be more funding for, biomedical research on other emerging pathogens. So hopefully the public understands sort of the process a little bit more than maybe they did before, and that will help with the process in the future, I hope.
I'm going to be a little bit more broad in answering the communication part of that question. I think it's really important for the general public to realize that science is very, very methodical. We don't just make up things on the fly. We're going to have, there's going to be a lot of what appears to the public to be confusion when results don't necessarily confirm or match. But that's part of the process. That's part of the process of science. Science is self-correcting. It's about making a hypothesis and proving it or disproving it. And so I think that can be frustrating to the general public quite often. It makes it look like scientists don't really know what they're doing, but in reality, it's the process just working itself through. For COVID-19 and SARS-CoV-2, it's not going to be an overnight eureka moment. This is going to be something that takes months to years to really resolve. We may have some drugs that work really quickly or a vaccine that works really quickly, but this is something that's going to take a long time to really solve the questions that we've been talking about today. And that certainly will impact our treatment strategies moving forward and vaccine strategies moving forward. This is just something that's going to take time, and I want the general public to understand that this is just part of the process.
And continuing with communicating to the general public, what resources do you believe are the best places for people to find out what's happening? Just recently, I know I go and read scientific papers, but I know that not everyone will understand all of the terms used. So as far as great resources that are legitimate and accurate for the research going on now.
Normally, I would have just said the CDC or the federal government. I still will say that those are good places to go. Johns Hopkins University has run a really nice information site and center. I would still, for scientists or people with science background, certainly primary literature. But as James mentioned, the primary literature is full of SARS-CoV-2 SARS-CoV-2 and COVID-19 literature. I mean, how many vaccine candidates just did you mention? I mean, think about all the different papers that'll be coming out about that. So there's a lot of conflicting and confusing literature. But again, that goes back to my point that it's a self-correcting process, and things will begin to sort of work itself out. In terms of where to go for immediate information, not Facebook. That would be a definite no. Probably not Twitter. I would still recommend sites like the CDC, the NIH, certainly the NIH, and then some other well-respected universities are maintaining some really nice places for information. At least that's been my experience. I don't know about Nisha and James.
I'd say the one exception to Twitter was Lindsay Marr has a pretty nice Twitter page describing.
Absolutely. Yeah, no, there's certainly some good ones out there, but there's also some really bad ones out there.
Okay, great. So I know this was a question interesting to me as an up-and-coming researcher, but we know that biomedical researchers often have to shift their research focus based on current research needs. I know that personally that none of you were studying any of the coronaviruses before this pandemic happened. So how have you all applied your skills and training in other research areas in order to address changing with an emerging virus during a pandemic?
So mine was luck. I was preparing in February, and actually part of having you come on and one of my other graduate students come on or join my lab, we were gearing up to study influenza. And we had some grants that we were putting together, and this was in February. And so in February, I just did find and replace for influenza and popped in SARS-CoV-2 because some of the same questions we were about to start tackling for influenza seemed to make sense. But as you pointed out, and as the panelists have pointed out, nobody was studying SARS-CoV-2 before it really emerged in and around February. There are some investigators looking at SARS, the SARS-1 and MERS and some of the other coronaviruses, but SARS-CoV-2 is unique and it's a new pathogen. And so it gives us an opportunity to apply again our skills and background and resources to a new virus, which is horrible from a pandemic sense, but exciting from a research perspective.
And I will say as a virologist that while this is a magnitude that's unprecedented, certainly my career changed a few years ago. I was doing my postdoc at the CDC working on West Nile virus when Zika happened, and had a complete shift in my research program at that point. Obviously, it was in the same family of viruses as I was used to studying and the same mode of transmission. So there were a lot of similarities, but certainly that sort of like shift in focus of public health research is not new certainly to the infectious disease world. So to some degree, you have to just learn to be flexible and to remember what it's like to learn a system again, which it can be really hard. You know, I've done lots of animal inoculations before, but I had never inoculated animals the way that we have to for SARS. And it was frustrating to learn and to feel like I'm, starting over again in some ways. But luckily I've done this so many times that you can get through those troubleshooting, times and learn. And hopefully all of the experiences we've had before can influence, our ability to learn again in the future. And so that's, I don't know, it's probably going to keep happening as well. Mm-hmm.
Well, my very first, folks that know me know my story. My very first NIH grant was in 2009. I had a proposal studying influenza. And if you remember 2009, that was when H1N1 that pandemic happened. My proposal was originally not funded, but I got a call one day from a program officer at NIH that said they were funding me. And I said, well, okay, I'm working on my revision. It wasn't anywhere near, I think it was maybe even not discussed. And she told me, oh, you're in luck. We're funding every grant that has influenza in it. And so I gladly accepted, and it was really my first grant that was ever funded. So it happens. It has happened before, and it will happen again. And so I think that's why it's important. What we're doing now sets a precedent for how we handle these things when they happen again.
Yeah, so I have a similar story to Nisha that during the Zika outbreak, we had to shift gears. I normally work with viruses that are from a different family, so it was a bigger shift for me. I mean, I think people assume that virologists know everything about all the viruses, but that's not really how it works. You kind of know a lot about the viruses you work with, and then you have to go back to the textbooks when there's a new virus that comes out. So with SARS, especially being that it's this massive virus that has a lot of different genes and a kind of a strange system of producing its proteins, it was a lot of kind of when it first hit, kind of learning how the virus replicates, how the virus produces its proteins. But other than that, we could apply a lot of the same techniques that we use for flaviviruses like Zika or alpha viruses like chikungunya virus. So kind of adapting those systems and updating them for this a little bit more challenging system.
Great, thank you. So now we're going to move into some really great questions in our chat box. So starting with our first question for our panelists, are there any studies on long-term health abnormalities or risks in fully recovered patients along similar lines as to how chickenpox infection causes shingles later in life?
So at least from my perspective, I think it's still too early on, right? I mean, we're only, 9 months, 10 months into this. There are some studies that are that are done that are more longitudinal. At least what I'm aware of, at least the things that I study and the things that I follow in terms of the immune system, the immune response, those types of things, really, there's not enough data, at least for me. I don't know about Nisha and James, that's the same case for them.
Yeah, I mean, I think one big difference here is that with shingles and chickenpox is that virus becomes latent in the system and then reactivates so that you're persistently infected with that virus, whereas your body clears this virus. As far as we know, like Coy was saying, maybe there's some other RNA viruses can do something similar, but not quite like herpes viruses, like chickenpox. So it's unlikely that the virus would reactivate to cause later problems, but it could cause long-term effects in terms of how it affects your lung health and other diseases that it can cause.
Yeah, the inflammation that happens after you clear the virus, this inability to really clear the inflammation, the inflammatory response is really vigorous, and that can cause a lot of collateral damage. Your immune system's trying to clear this virus, and in the meantime, it damages your lungs. You get a lot of lung remodeling that occurs. There's a lot of buildup of basically, for lack of a better term, there's a lot of buildup of things like mucus and excess. You really lose a lot of lung function, and that can have lingering effects. You're certainly seeing reports of people suffering from just due to the loss of lung function. You're seeing studies or results of people that have lingering conditions or lingering effects for 4 to 6 weeks. That's very different though than really how I'm reading the question is in terms of much longer term effects. I would still consider that just part of a short term effect. I mean, really, you're looking at 4 to 6 weeks worth of clinical course.
I think with some more time, I'm sure we will learn more about the long-term impacts.
Okay, so one of our next questions in the chat, which I think you, Dr. Allen, will have some good insight on, is there anything we can learn and apply from vaccine production post-2009 swine flu epidemic?
Well, the way that vaccines are being made now are very different than what you have to do for influenza vaccine, right? So a lot of influenza vaccine is still made in egg, and so it takes it's a much different process. And actually, I'm going to punt to Nisha or James on this one because the technologies that are actually being used now, I mean, they all have grown. Vaccine production has been really unchanged for centuries until the last, say, 20 years or so, with really the rise of genetics and the rise of biotechnology, providing us with new resources to develop vaccines. So we've always been learning from vaccine development, but at least in terms of influenza, I think the vaccine process for SARS is drastically different than what we use for flu. Oh, I just get nods from the virologist. All right, cool.
No, that's correct. I mean, yeah. It depends on the type of vaccine. So it's really, there's so many different types of vaccines being produced that it's difficult to say. I mean, but they're not being produced in eggs, that's for sure. They're either, if it's the plasmid, it's being produced in bacteria. If it's a viral vector, it's being produced in cell culture and in giant bioreactors. So the bioreactors, excuse me, these are processes that have been developed for other vaccines that are produced in cell culture. So they should be able to adapt them relatively easily to these types of vaccines as well. So I think that they should be able to generate a lot of vaccine in a short amount of time due to the research that's been done for other vaccines previously.
I know that many people are curious with the vaccines, if we're going to have a vaccine more like a flu vaccine where we would want to get it yearly versus like a one and done vaccine with a couple of boosters, like when you're a child. What do you guys think about how vaccination is going to happen in the future?
Just based on the initial results, my feeling is we're going to have to be boosted quite often. But again, it's still kind of early on. We don't know how effective. It goes back to one of my earlier questions or one of our earlier discussion points. We don't know right now how effective or how long lasting that immune response is going to be. And that's going to dictate how many times or how often we have to get vaccinated.
I think for influenza, like we'll learn a lot about how the virus evolves throughout a season. And that's what sort of drives the need to update the vaccine every year. And obviously if there's, with influenza, there's new strains that we haven't seen before that get reintroduced, then we really need to update vaccination. So it kind of depends on how things go with the evolution of the virus as well.
I was going to ask you, Nisha, well, I mean, how fast is it evolving? Is it a fast evolving virus?
From my understanding, it's not like flu, sure. But James, you have some thoughts on that?
Yeah, I mean, if you look in the news, there are apparently a new variant arising quite often, whether or not it's just kind of a random event or whether or not there's actually changes that are being selected for because there's more human-human transmission or whatever. It's too early to say, really. But this virus has the ability to kind of fix any mistakes it makes when it's replicating. So it appears that the mutation rate is not quite as high as some other viruses, especially not with flu where they can reassort and create genetic diversity like that. So I think the bigger issue is whether or not we actually develop a strong protective immune response that lasts over a long period of time.
I agree.
And continuing on the lines of flu, we have a question in the chat about how do you think the SARS-CoV-2 pandemic will affect the incoming flu season coming up?
I think it's a double-edged sword. I think we are as hygienic of a society as we've ever been at the moment. So I think that will help. The problem, though, is having two diseases like this circulating at the same time. Honestly, I think it's going to be a really bad flu and COVID-19 season. But we are much more hygienic. I mean, I haven't used this much hand sanitizer. I mean, it's amazing how much hand sanitizer my family goes through right now. And we're all wearing masks. We're all trying to socially distance. And so all of those things will help keep us safe from flu. The problem, though, is if we get flu, will we have the medical resources available to us with all of these being occupied by SARS-CoV-2 and COVID-19 patients?
I think diagnostics and therapeutics and treatments are also a concern. I think most of the diagnostics for influenza have been adapted to include SARS-CoV-2 so you can diagnose the difference between the two. And certainly, of course, there are some antivirals for flu, which could be effective, particularly, disease could be worse in folks who are co-infected. It's hard to say what would happen.
Yeah, co-infection with flu is typically flu. One of the frightening aspects of flu is the ability to get co-infected with other agents, and oftentimes the morbidity or the mortality rate we see with flu is actually due to a co-infection with another virus or bacteria. And so there's certainly the possibility that you could get infected with something else, such as SARS-CoV-2 when you have flu, and then that would be probably a really bad outcome.
So a general plug to get your flu shot this season.
Also, yes, a general plug to get your flu shot this season.
So moving away from flu, our next question from the chat is, to what extent do you see misinformation and mistrust of science in the general public as a failure of public outreach in biomedical sciences? And what do you see as the solution to this?
Well, you have a speech.
Yeah, I mean, I think clearly with the access to information that is present on the internet is also the access to disinformation. So because people have access to YouTube videos where people have their opinions and don't have to back anything up, you can just spread whatever you want, whether it be about flat Earth or any other conspiracy theories. And so because people don't have that background scientific knowledge, they're open to believing what other people say if they seem legitimate. So I don't really know what the solution is, because people don't trust us as scientists, so they're not going to listen to what we say. So, I mean, I think better education in elementary school, middle school, high school, I think that would probably be the best start, is making sure our kids have the best education so that when they're adults, they're not as susceptible to misinformation.
Yeah, I think the accurate information is out there. It's the ability to determine what's accurate versus inaccurate is really the problem. And the amount of misinformation, both accidental and purposeful misinformation, is a major issue. And so, to be honest, I think it's not an issue with us in the biomedical sciences. We're doing our jobs. We're getting the information out. It's the amount of misinformation that drives mistrust, and a lot of that is absolutely on purpose for different agendas and different gains.
I think having clear messaging from at all levels too is important. And so sometimes, mixed signals from even from people who are in positions of respect can be confusing. So, I mean, I understand how people are confused.
Yeah, I think events like the one that we're having right now, which can communicate and people can ask questions about what they don't understand, I think things like this would be really great in the future, and hopefully it can be a recurring thing as we keep finding out more information. Our next question in the chat is, do you think behavioral changes adopted to decrease COVID spread will impact development of the immune system in children? I mean, I know you've heard the rub some dirt in it, get your immune system activated, but in this case, that might not be the greatest, so.
Rub some dirt in it and get your immune system activated. I love it. I love it.
Isn't that what you guys are studying, Coy?
Yeah, well, yeah, pretty much. Yeah, I mean, so certainly the behavioral changes, right? So having kids myself, all right, and that is what I usually do. Just rub some dirt in it, you'll be all right. I think the behavioral changes that my children have seen us go through. I mean, right now they a lot of times they'll remind me to put my mask on, and a lot of times they're the ones to be the first to show up with the hand sanitizer. They're talking about it in school. It's impacting their life, and so I think. This is really ingraining a sense of behavioral change in the kids. And I think that will translate to them kind of as they grow up. They'll always be able to look back at, you remember that pandemic year we had? And so, I really think that this is really setting up a system for behavioral change. Already in Asia, you see way higher mask usage At the same time, you see lower rates in many cases, be it SARS or other diseases. And I think that's a behavioral acceptance there that wearing masks, for example, is okay. And I think here in the U.S., it's been slow, but it is coming around. I think a lot of these changes are beginning to take hold. And I think ultimately using sanitizer, using masks, socially distancing, as we see it working in other places, hopefully it will be wider adopted here. And I think ultimately this will help not only the development of kids' immune systems, but ours as well. Our immune systems are always evolving, they're always adapting, they're always developing. And so I think across the board, all of these behavioral changes will benefit.
But Coy, do you think this could lead to higher rates of autoimmunity or things like that if kids aren't getting exposed to normal flora bacteria because they're killing all of it on their hands and whatnot?
We already have, not necessarily that, the hygiene that we currently have in the United States is driving a lot of that already. And I actually have a great case in point I use in a lot of my classes where I had one of my daughters that was born and took lots of antibiotics all through her childhood for ear infections that changed her microbiome, and now she has lots of allergic issues, skin disease. I should say skin disease. She's got like psoriasis and some dermatitis issues, but she's also way more prone to develop respiratory, like hay fever or pollen allergies. Versus my younger daughter, which is never had any problems at all and is still a champ all the way through. I wish I had her immune system. And so yeah, I think the hygiene that they grow up in, what they're exposed to as children will absolutely drive a lot of these effects that we see with their immune system development over time. But we're already there. We live a much more hygienic lifestyle than our ancestors ever did. And you are seeing rises in allergic diseases and other diseases associated with us having just a cleaner lifestyle.
So we have one last question to kind of tie the whole talk together. And how do you see the field of biomedical research changing as a result of the COVID-19 pandemic?
Everyone's going to become a virologist, I think, is what's going to happen.
Well, and I think there's two sides of this. And so, yeah, I think there's certainly an appreciation for what we do in the biomedical fields. I think my kids and I think my wife now understands way more about what I do because of all the work and meetings I've had at home just with her sitting in the background. And so there's certainly that increase in appreciation that occurs. And you know, I think over time, this is just going to, you know, I think what we learn now is going to be applicable later when this happens again. And so I think biomedical research is always going to play a role as when any disease emerges or anything like this happens again. And my hope is that we learn from this and can apply what we know when this happens again.
It has to get better.
Yeah, it has to get better.
Okay, well, thank you so much to all of our panelists for being here and speaking on biomedical research and answering some really great questions. I'm sorry if we didn't get to your question in the chat, but I know that all of the panelists we have here will be happy to answer questions by email and share their knowledge.
Absolutely.
Sure.
And just wanted to thank everyone again for joining us here today. For those of you watching, we do have 2 additional webinars this week. One is tomorrow night from 6 to 7:15 p.m. So if you need a break from election night anxiety, feel free to join us. We're going to learn, we have 3 speakers, one from the field of public health, one who's a pediatrician, and one who is a practicing veterinarian, who are going to talk to us about how COVID-19 has sort of impacted the work that they do, the people they work with, as well as them personally, because we know this has been a strain on everyone. And then we have another talk on Wednesday night, same time, 6 to 7:15 p.m. And we're going to be talking with the leaders of 2 different organizations, Virginia Harm Reduction and the Street Dog Coalition, to talk about how COVID-19 impacts those who are working with vulnerable populations in the field. So we'd love for you to join us on that. And again, I'd like to thank our speakers and our lovely moderator, Juselyn Tupik, for joining us today. Thanks, everyone.
Thank you.
Thanks.
Thank you.